Good morning, everybody, and thank you for getting up so early. I always feel like a bit of a fraud. By the way, this is where I live now. This is Okinawa. It's a seaweed factory, as you can see, or a seaweed farm, I should say. And I like to walk with our dog, Momo. About half the female dogs in Japan are called Momo. It means peach. Anyway, so Momo and I walk around here, and I used to let her run free off the leash until one day she ran away, which was very embarrassing. She actually ran home, so that was good. But then I discovered that it's against the law to let your dog run free in Japan. And so from now on, I keep her on the lead. But it means that I walk for almost 2 hours a day with this wretched dog, who really belongs to my wife. Anyway, so, you know, you're looking at an extinct volcano or possibly a living fossil, because I haven't had a lab for a long time. So what I'm going to tell you about is the story of my young life when I was more like you than I am now. So I grew up in Oxford, and I was surrounded by... Oxford is the home of lost causes and also of... you know, my dad was an expert in paleography. His great moment of triumph was when he recognized the handwriting in a manuscript in the Bodleian's collection as belonging to that of the poet Petrarch, because he knew what Petrarch's handwriting looked like, and he recognized it. So that was a sort of enormous discovery. So most of his friends were medieval historians. And I was, A, no good at history. I always got confused by the stories. And B, it required a knowledge of Latin and to some extent of Greek, at both of which I was increasingly useless as time went by. So from quite a young age, I wanted to be a scientist, and this was very much encouraged by my first science teacher. Here he is, he's no longer with us, Gert Sommerhoff, who was a young German who really made science interesting and fun and clear. I mean, some of his theories were, I think, outrageous and wrong. For example, he said, the reason why blood is red is so you can tell when you're bleeding. Well, I mean, that's a handy byproduct of the fact that blood is red, but I don't think it's the reason why blood is red. I mean, we'll get to that in a second. But I really look, we had one, whereas we had, I guess we must have had 12 Latin lessons a week, and we had one science lesson a week, and I really, really looked forward to that one science lesson. And then subsequently, I've always been rather keen on reading scientific biographies, and the first one I read was that of Madame Curie, which is an inspiring story. I don't know if probably some of you have read it. It's an absolutely amazing story. I mean, she was such a determined woman, and she won 2 Nobel Prizes, was involved in a sort of sex scandal, and it was, you know, very, very, very heroic. And so that was an inspiring story. And then there were various other things which helped encourage the young scientist. One was that there were Christmas lectures in the local museum, and they were great. You know, people did experiments before your very eyes and made explosions, or froze things in liquid nitrogen and showed they went brittle and things like that, and they were very popular. And then there were lectures after school, and one of them was by somebody from the biochemistry department. And by this time, I had a very good chemistry teacher at my secondary school. And I forgot to mention that Gert it was who taught me that I was a good biologist, because I did very well in a biology exam at the age of 12 or so, and I came out very near the top of the school without really trying. So I sort of realized that my mind worked in biological way. But I was also very keen on chemistry, and when I heard these lectures on biochemistry, I mean, this is appallingly complicated, and actually, on listening to him, the question that came in my mind was what controls all these reactions? I mean, it's all very well to say this goes there and that goes there, but what controls the flux of these chemicals? And actually, at the time, that was a very good, a sensible thing to think about, because it really wasn't known. And very excitingly, when I was an undergraduate in the early 60s, people began to figure out how biochemical reactions were controlled, sometimes by increasing or decreasing the amount of the enzyme, or by altering the enzyme's activity by something that a French scientist called allosteric. So I knew that, you know, there was this subject, biochemistry, and I was really terrifically lucky, because it was a wonderful time to get in on the ground floor, as you will see in a second. Now, because I lived in Oxford, and my parents were all, well, my mother wasn't academic, but my dad was academic, and I knew it was important to get away from home. And more importantly, Cambridge was the place to do science. It is actually still true, I believe, that 2/3 of the people in Cambridge are scientists, whereas 2/3 of the people in Oxford are not. I'm actually rather an expert on the differences between Oxford and Cambridge, and although people tend to sort of lump them together in one, they're actually unbelievably, in every particular, they are different. Very different ethos and different traditions and different ways of doing things, different governance, and so I went to Cambridge. And of course, Cambridge did have this wonderful, I love this. This is Alexander Pope. Nature and nature's laws lay hid in night. God said, let Newton be, and all was light. And so one was sort of in that grand tradition. One didn't remotely suppose that you were a second Newton, or even a sort of micro-Newton. But nevertheless, you know, that was the example. And I used to walk to work through the old Cavendish lab. It was no longer the Cavendish lab. That had moved out of town to larger premises. But one walked in a space of about 50 yards past the place where Maxwell had worked out his equations. Thomson had discovered the electron. Rutherford and his boys discovered the atomic nucleus and the proton, the neutron, and first split the atom. And that was pretty amazing. You know, here you were, just a humble graduate student with these amazing role models. And then, of course, in that same department, Francis Crick was working, and Jim Watson joined him. And together they worked out the structure of DNA. Actually, they didn't work out the structure of DNA. The structure of DNA had been determined by Alex Todd and, oh gosh, what was he? Brown. I've forgotten his first name anyway, very nice man, who worked out the chemical structure of DNA. That was really rather important and was essential information. And Lord Todd, we used to call him Lord Todd Almighty, because he was a very grand man indeed, said, I was the one who worked out the structure of DNA. Watson and Crick merely determined the configuration of the two strands. And I guess he was right. Anyway, here they are. And I did well enough in my exams to, you know, be accepted as a graduate student. So, big problem is, what problem do you work on? And I think this is the biggest problem that any scientist, or I was discussing with the next speaker, any mathematician has. You know, what do you work on? So how did I find a problem? Well, the first problem I found was my supervisor said to me, Tim, why don't you go to the library and find a problem? So I went to the library and I found a problem, and it was a good problem. As far as I know, it's a problem that still hasn't been solved to this day. And fortunately for me, it didn't work. It involved killing rats. And I'm not sure who liked it least, the rats who were being killed by me or me killing the rats. But in either case, it was unpleasant. And luckily for me, I hadn't read the materials and methods carefully enough. And I was using the wrong kind of rat. So the experiment didn't work. So after about 6 months, I had complete failure. I mean, just nothing worked at all. And I went to my first scientific meeting, and I heard a talk from this guy on the left and also the guy on the right, which really sort of set my course for the rest of my life, amazingly enough, although I didn't realize it at the time. So you see, here's Borsuk. As far as I can determine, this is the only account of that meeting that exists. He wrote a review. It's a very strange review. Early development of the echinoid egg, that means sea urchin eggs, compared with erythropoiesis. Now, sea urchin eggs are at the beginning of their life, and erythropoiesis means making red blood cells, which are at the end of their life. They're destined to die. And what do they have in common? The answer is absolutely nothing, except they're both round. Nevertheless, he'd worked on them, so he talked about them. So the first thing was that in sea urchin eggs, protein synthesis increases after fertilization. It turns on. So that was interesting. And then he'd also studied the business that if there's an interesting hemoglobin, the stuff that makes your blood red and carries the oxygen, has 3 components. 2 proteins, the alpha and beta chains of globin, and the prosthetic group, which makes it red, heme, which has iron in it. And it had been discovered that if you deprive these immature red blood cells, which are actively making hemoglobin, of iron, that was the easiest thing to do just by adding iron chelating agents to the medium, they stopped making the protein. And that was kind of interesting because, you know, the heme is made in the mitochondria. It's completely different chemically from the protein. There's no reason to suppose, except that, of course, you can, it makes evolutionary sense because if you make a lot of protein without the prosthetic group, the globin is terribly insoluble and precipitates out and would, you know, just like sort of neurofibrillary tangles. It would, or sickle cell anemia, you know, that's globin precipitating. And so that was very interesting. And then the question was, when does the heme actually enter the globin? Do you have to make the whole thing and then put it in, or do you make it while it's on the fly? So here is a picture. This is not actually hemoglobin, it's actually myoglobin. This pointer, boy, oh boy. They never have industrial strength enough pointers, you know, for these back-projecting things. You really need something much, much more powerful. So to go and sort of point. So here's, I love this. This is the sausage model of myoglobin. It was Kendrew's first. It really annoyed Max Perutz, I think, because Kendrew got there first. And you can see the heme, the red heme, nestling in a fold of the protein. So Ingram, that's the guy on the right's talk, presented evidence that the way this thing worked was that as you made the globin change, so here's messenger RNA, the blue line, with ribosomes translating it. So you have to imagine I'll be a ribosome. I start at the beginning of the message, and the protein sort of comes out from here. And I get to the place where the heme belongs, okay? And if there wasn't any heme, his data suggested that the ribosomes then stopped and waited for the heme to arrive. How they knew that there was no heme, he didn't, you know, the data suggested that. So boy, what an interesting idea. So went back to the lab and excitedly told my fellow graduate students about this. And as we worked out what the data actually meant, we realized that Ingram had got it wrong. I mean, 180 degrees wrong. If anything, it suggested that in the absence of heme, the ribosomes sped up or something. And then, and this I really can't reconstruct, we decided that we could do the experiment in a much better way anyway. And with the arrogance and innocence of youth, decided to devote ourselves. This was a really good problem. How did the heme control protein synthesis, and when was it put in? Did the ribosomes actually form cues? And so my PhD was greatly facilitated by my first benchmate, Lou Reichhardt, who was also interested in this problem, and amazingly completely failed to make any progress. Well, not amazingly. It was difficult to dissolve the heme, for example. There were various reasons. But he knew how to make these immature red blood cells, the blue cells here called reticulocytes. And it's very easy to make a rabbit anemic and to have most of its blood be these reticulocytes. And then later on, it turned out to be very easy to get the guts out of these cells. All you have to do is to purify the cells, which you can do easily by repeated centrifugation and washing. And then you just add distilled water, and they break by osmosis, and the cytoplasm all comes out. And actually, that works just as well as the cells. So this was a great advance. And then the other person on the left was Tony Hunter, who joined the lab a year after me. And Tony and I basically sort of collaborated on our PhD and taught ourselves how to do the experiments. And they worked very nicely, and we found that actually the ribosomes didn't form cues unless we specifically asked them to, and we had ways of showing that our methods would actually demonstrate the existence of cues. But normally, if you starved the cells of iron or heme, there were no cues. So Ingram was utterly wrong. I mean, just completely utterly. And that was cool. Now, in the middle of all this, so this would have been in 1966, I went to my second scientific meeting, which was also very important, because I met this man on the left here, Irving London. And Irving was interested in exactly the same thing that I was. He'd actually worked with a guy called David Shemin, who'd worked on the biosynthetic pathway of Heymann. And so we were both really interested. And it's sort of interesting. It sort of exemplifies something that has been my experience. Although there was a great disparity in our ages, I mean, Irving was probably 20, 25 years older than me, and he was actually, as I didn't realize at the time, the chairman of the Department of Medicine. But because of our shared interest in this problem of how the heme controlled globin synthesis, we became really very good friends and collaborators and colleagues. And I actually went and worked in his lab. And, you know, we used to have, he sent his folk off to Cape Cod in the summer, and it was hot summer in New York, and we used to have dinner together and stuff. And it was really, really nice. And scientifically speaking, you know, we were on absolutely level pegging. And so when I came time to do a postdoc, there was no question. I went back to his lab and tried to study. Now, by this time, we got the cell-free system working, and the cell-free system on the right is an experiment that I must have done long, long ago, showing that you can see the general thing. You're measuring time along the bottom, radioactivity and protein up the side, and if you add heme to this extract, protein synthesis carries on at pretty much at the rate that it would have been carried on inside the cells, although there are no cell membranes left and no mitochondria or anything like that. If you leave out the heme, protein synthesis starts off all right and then curls over and dies. It doesn't die completely. It typically goes to 5 to 10% of the initial rate. And if you add back the heme, it recovers. So there's a reversible, whatever the control is, it's reversible. Now, at that time, we really didn't know how things worked. And all we knew was that if there wasn't any heme there, the ribosomes couldn't get onto the messenger RNA. There was a block to the process of initiation. But we didn't understand the process of initiation. And actually, it was my friends back in Cambridge who made a very important discovery, which the chains always start with a special transfer RNA. All the proteins start with methionine delivered by this thing. And I was very envious of their findings because that was a really important advance. And I was just sort of jiggling around. In fact, I mean, you know, I really made no progress in the understanding of this whatsoever. So making no progress is depressing. So what you do, at least what I did, was to sort of start trying little side projects. And one of the things that I decided to do was to map the genes in poliovirus, because the methods that I'd used for the globin would have worked for poliovirus. So I got some poliovirus from Ellie Ehrenfeld, who was on the 3rd floor. I was on the 6th floor. and added it, and it didn't get translated. And I thought, well, there's a good reason why it's not getting translated in these rabbit red cells, because they don't normally get polio. So what we need is some extract from a polio virus infected cell. And if we add that, it must have the factors that you need for translation. So I did this and got the astonishing result that when you added poliovirus infected cytoplasm to the rabbit reticulocyte lysate in the presence of lots of heme, you got a shut-off of protein synthesis that looked exactly like you left the heme out. So what the hell was it? So we then tried to find out what this mystery thing was, and the extraordinary thing was very large, and it didn't go away if you digested it with DNase or RNase or protease, so it seemed to be some very mysterious big thing. It did go away if you treated with alkali or acid, so that was satisfactory. And these were sort of naughty experiments. They weren't part of, I had to do them after the technician went home in the evening, so, they were sort of late night and Saturday morning weekend experiments. And it took about 6 weeks before we realized that actually we'd been misled. This happens so often, We were totally misled because of the RNase result. Actually, this was RNA, but it was double-stranded RNA, which is highly resistant to digestion by pancreatic ribonuclease. So once we discovered that, everything was clear. But then this is weird. How come double-stranded RNA inhibits protein synthesis just like leaving out the heme? This makes no sense at all. And then I discovered another thing. We had an Israeli woman in the lab, and she was working on glutathione. And we discovered that tiny amounts of oxidized glutathione caused exactly the same kind of inhibition. Again, completely different. Weird, weird, weird. So that was very good, and so it set the scene for my return to Cambridge. I mean, it really wasn't a very successful postdoc. I hadn't published very much at all. But I had discovered this interesting double-stranded RNA effect and the oxidized glutathione effect. So I went back and collaborated with this guy here, Richard Jackson, and who is much cleverer than me, and we made a very good team, because I sort of like broad brushstrokes, and he was more concerned with the detail and very highly intelligent. And it was, again, a lovely collaboration, because we sort of were absolutely in tune with one another. And we discovered, by using the discovery that Richard and Tony had made, that when you labeled these things with methionine, this is really sort of too complicated to. This is a sucrose density gradient, and this big peak here is single ribosomes, and the single ribosomes, as you'll hear from Ada, are made of two subunits, a small subunit and a large subunit. And you see this dotted line here is radioactivity, and that radioactivity is in methionine, and what there is, the 40S, the small subunit, actually has initiated tRNA bound to it. And it's actually that complex of the initiated tRNA bound to the 40S ribosomes that goes looking for the start of the message. Now, this was completely against the idea. Everybody knew that the ribosomes only bound tRNA when they had messenger RNA, and these complexes didn't have messenger RNA. And we had a lot of trouble with the reviewers, but we were right. And they were wrong to be skeptical, because our evidence was very good. So that was tremendously important. That was the step that was inhibited, the formation of that pre-initiation complex. And then the lab burned down. And it was the best thing that could ever have happened to us, because at the time we were distinctly foxed by what was going on. Fortunately, the two graduate students had just graduated and done their thesis. I mean, absolutely nothing was left. Nothing, that is to say, but a hydrogen cylinder, which is arrowed on the right-hand side, and our liquid nitrogen refrigerator. So all the most precious samples were safe, although the nitrogen had melted its seal, so it was venting nitrogen at a fantastic rate, but it was easy to find somebody who had a spare, even though it was a Saturday morning. And, of course, we had to move, because our lab was utterly destroyed, and we moved up the road to a hospital lab, a teaching lab in the hematology department, ironically enough, It was great because the university had a wonderful insurance policy. We got all new shiny equipment. And perhaps more importantly, Max Perutz, who was then the chairman of the famous Laboratory of Molecular Biology in Cambridge, very scary place, said that we could use his stores and use his tea room. And here it is. We were sort of working somewhere in Addenbrookes Hospital. There, up there is the famous lab, and just had to walk across the road. And the tea room was full of these giants of molecular biology. And it was amazing. You know, Francis Crick would come and sit down at your table and explain what he was thinking about nucleosomes. And that was absolutely great. And, you know, Fred Sanger was very kind to me. Fred, if you met a double Nobel Prize winner and an extremely wonderful, wonderful man. Very modest. If you met him, you'd think he was the janitor or the gardener or something like that. You had no idea how brilliant he was. You know, so this was, again, a fantastic. I hung out mostly with John Gurdon and his students and postdocs. So I was very interested in developmental biology. And I think the interesting thing is that there are all these sort of elder statesmen who we didn't think we could possibly emulate. But there were all these youngsters who were sort of roughly my age, and we chatted to each other, explained what we were trying to do, you know, and criticized and helped as much as we can. We worked on a wide variety of different problems, and it never occurred to us that, you know, we were sort of future Nobel Prize winners. It was just sort of, you know, you just put one foot in front of the other and thought as much and worked hard and stuff. It was no big deal. It's sort of interesting. Anyway, I think largely as a result of this sort of purification of the lab by fire, we came across the answer. And the answer was that we were dealing with one or more protein kinases, enzymes that stick phosphates onto other proteins. And this is the graduate student Paul Farrell's sort of demonstration publication gel. And again, it's really feeble. This doesn't, I don't know whether its batteries are failing. Let me try, I've got another one here. My one is not as powerful, but I think it's working better. Boy, I can't see it at all. And if I can't see it, you certainly can't see it, so we'll forget that. Anyway, that band there is P32 label. So the way it works is that in all these conditions, an enzyme turns on that phosphorylates the initiation factor that binds initiator tRNA to the 40S subunit. And that's what, that was the answer. And so here's an example not of inhibition, but of activation. You stick that phosphate on to that protein there, and this guy is active, actually. It's about 100 times more active than that guy. If you look at the structures, they look amazingly similar. I mean, it's extraordinary how tiny changes in the configuration can make a huge difference to the biological activity of these things. And as the biologists among you will know, it's, you know, practically everything you do in cells is controlled by reversible phosphorylation. Every time I move my arms, every time I have a thought, phosphorylation is going on. And it turns out now, years later, I mean, this is not my work at all, that there are 4 of these enzymes, and they detect various conditions in which it's desirable to shut off protein synthesis, and I'll say no more about that. So the double-stranded one is the one at the top. It's now called protein kinase R, and it's a heme control. And we won't go into that. So we were pretty pleased with ourselves because we've been working on this for, I don't know, almost 10 years and solved the problem. And it feels great when you solve the problem. And the problem had a nice clear, so that was why it was a terrific problem. You know, it was very clear-cut, and it had a very clear-cut answer. I mean, lots of details to be filled in, of course. So we organized an EMBO workshop, and one of the people that we invited to the workshop was Tom Humphreys, who worked in Hawaii, among other things, on the control of protein synthesis in sea urchin eggs, because I remembered that thing, and I was now, you know, the problem is when you solve a problem, you don't have a problem anymore. And you have to, it's just like going back to graduate school. You've got to find yourself a new problem. So I thought it would be fun to look at these sea urchin eggs, but there were no sea urchins in Cambridge, and I wanted to know what the latest was. So Tom and I became friends entirely because of a shared love of cycling. So he wanted to go for a cycle ride. I had a bicycle. I lent him my bicycle, and on that basis we became friends. And I didn't know this at the time, but he was a director of a summer course in Woods Hole. And he said, well, how about you come to teach the course next summer in Woods Hole? And we can do some experiments on sea urchin eggs. So that's what happened, and I did, and it was a great move. But again, that first summer, it takes a while to get familiar with the systems, and sea urchin eggs are a terrible thing to work on because you only have them for at the most about a month of the year. Here they are. This is how you get them. You give them a 12-volt alternating current electric shock, and the female sheds her eggs. Do the same to a male, and the male sheds his sperm. Sea urchin sex can't be very exciting for a sea urchin, I suspect, because they don't come into contact with one another. The gametes go into the water. So it's really easy to fertilize sea urchin eggs. You just take some of that sperm, dilute it to the right concentration, add it to the eggs, and they get fertilized. And here they are. But there's one very important thing about that. So this is a movie. So I add some sperm to these eggs. There's one non-egg up top left, by the way. That's an oocyte. And you see that this is highly speeded up, almost 1,000 times. They all divide incredibly synchronously, completely naturally. You don't have to do anything. They all divide within 5 minutes of one another. First division after about an hour, subsequent divisions every half hour. So that's really important. Now, I went back again in '79, the second summer, and heard a beautiful, beautiful talk by this guy, John Gerhart. And he described what happens when you add progesterone to frog oocytes. Now, it's important to, and this is something I didn't really get for a long time. So what is an oocyte? Well, the answer is that all the females in the audience of reproductive age have oocytes in them. And in humans, the oocytes get ready, turn into eggs once a month, and they can be fertilized. In frogs, however, the female frog is full of oocytes, which mostly get shed in the spring. And the males clasp the females and dribble sperm out over them as the eggs come out. But in order for the oocyte, which is the precursor of the egg, to turn into an egg, it has to undergo a process called maturation. Various changes to the surface of the egg and things going on inside, which needn't really concern us here. And this is triggered by progesterone. So again, this is a movie, so watch carefully those 3 oocytes on the right-hand side. Not very dramatic. A white spot appears, and then there's that little convulsion at the end is actually what's called the first, sort of first meiotic division and the formation of the first polar body. And what John described was that a Japanese called Yoshio Masui had discovered that what triggered this change, the formation of the white spot, and if you look underneath the white spot, you see chromosomes lined up on a metaphase spindle, was something he called maturation promoting factor. And basically what it did was to trigger a cell cycle transition. So beforehand you have an oocyte with a big nucleus and you can't see the chromosomes. After adding progesterone, you see the chromosomes condense, the nucleus dissolves, the contents of the nucleus mix with the contents of the cytoplasm, the chromosomes all line up. And if this thing was to be fertilized, the chromosomes would come apart and you get the first tiny cell polar body and the egg, and then the second meiotic division. But what Masui discovered was that if you suck out a bit of cytoplasm from the mature oocyte, matured oocyte, you call it an egg or an egg, and inject it into a second oocyte, no progesterone now, this transition occurs very rapidly. There was something there, and that something was clearly an enzyme because it could digest it with proteolytic enzymes, and it was heat labile and so forth. And John described the experiments that he and his colleagues had tried to characterize this amazing material. I mean, it was pretty magic stuff. I thought, gosh, what a great problem to find out what MPF is. And they didn't really succeed. They didn't make any progress on that, except to find when you found it and when you didn't. And this is a sort of diagram of when you find it and when you don't. It comes up as during maturation on the left. It then stays up until the sperm arrives. And then it comes back for each division. You only find it in dividing cells. You do not find it in between. So, and then it was discovered that this stuff was not just something in frogs, but you could discover it in starfish and even eventually in human cells. So it was sort of universal. And frog MPF worked on humans, and human MPF worked on frogs. So, whoa. Let's see if this, I don't know how this works. On. Oh, yeah, look at that. Can you see that? I can see that. Cool. So I started thinking about this, and there's a lot of other stuff that made me wonder about cell division. And one of the important things that I haven't mentioned, you remember I said that protein synthesis was activated. We made no progress in finding out how protein synthesis was turned on, but I became deeply aware that if you inhibited protein synthesis, very easy to do with antibiotics, the cells did not divide. They went through their first round of DNA synthesis, but they couldn't divide. So one or more newly synthesized protein was important for cell division. And I was thinking about MPF and all that stuff. And I then did an experiment, and the experiment was actually designed to see whether the patterns of protein synthesis in parthenogenetically activated eggs were the same as or different from the patterns of protein synthesis in properly fertilized eggs. So I did an experiment, and the experiments are always the same. You add some radioactivity, and you take samples every 10 minutes, because you might as well take samples. And looking for the pattern changes, you know, you didn't know when they were going to change, and it was boring to sit there waiting for an hour. You might as well do something while stuff is going on. So I took samples, and this was an unbelievably simple experiment, which anybody in the preceding 10 years could have done, but didn't, I suppose, because they didn't think it was worth it. But I did it, and you can see what I discovered. I discovered that the first protein, really abundant protein, here it's the strongest protein, came and then disappeared, and then it came back again and disappeared again. And the appearance and disappearance was in time with the division. Actually, it went away about 5 to 10 minutes before the cells actually divided in two. And then an amazing thing happened, which was that I ran into John Gerhart at the wine and cheese party after the Friday night lecture. And I recognized him and went up to him and explained this result. And he, in turn, told me about a result that he and Mark Kirshner had recently got, which was that although the first appearance of this triggering here does require protein synthesis, progesterone actually triggers the translation of several new messages. This kind of transition, when you inject MPF into a thing, this does not require any new protein synthesis. But the next division does require protein synthesis. In other words, there was strong evidence that somehow something was degraded and you needed to make something afresh. And I'd just seen a protein that you had to make afresh. So I sort of knew that I'd completely accidentally stumbled on the secret of cell division. And we looked later in clam eggs, and we'd been working on these because these were interesting proteins for another reason. And you can see that in clams there are two of these proteins called A and B, which is how cyclins A and B. And I called them cyclins because they cycled and because I liked cycling. It was a sort of jokey thing, and nobody can stop you doing that. And it's a good name, and it's stuck. And we sort of worried about two things here. Number one was these proteins gradually accumulate, yet the entry into mitosis is a very abrupt switch-like process. So how can you turn something gradual into an abrupt switch? And second of all, was it really true that, I mean, these are 10 minutes apart. So here we are, we've completely gotten rid of the cyclin A, 10, 20, 30, 40, 50. You know, these are radioactive proteins. They're not actual things. So could you really make enough in half an hour to trigger cell division? So we did sort of back of the envelope calculations and thought, well, perhaps, you know, if it was an enzyme, you'd have enough. But it took a very long time, and I realized, to my horror, that I was a sort of expert on the control of protein synthesis, and I knew nothing about the control of the cell cycle, so one just had to bite the bullet and go back to graduate school and discover by chatting to people and reading what was known about the cell. And luckily for me, very little was known at that time. It had been discovered several years before that the process of division was separated from the process of DNA replication by gap phases, but what triggered these transitions from G2 into M, actually making a cell enter mitosis, or what triggered the separation of chromosomes at the exit from mitosis was totally, totally obscure. And the only progress that had been made was by a yeast geneticist, first by Lee Hartwell, who had discovered that there were genes that control cell cycle transitions, and he and his mates focused particularly on CDC28, just the 28th, the nomenclature here is terrible, because it controlled three very different processes to do with the start of the cell cycle. So they called this process that it catalyzed start, and they thought it must be regulatory, because how could one protein control three very different biochemical kind of things? And then Paul Nurse was inspired by Lee's work and decided to work on a different kind of yeast called fission yeast. And here, you know, if you take a temperature-sensitive CDC2 mutant, the cells can't divide, they just get longer and longer. So CDC2 catalyzes entry into cell division. And he also isolated another gene, which he called WEE1, because the cells were smaller, and he was working in Scotland at the time. And he realized this was very interesting, because this mutation, a recessive mutation, actually caused an acceleration of the cell cycle, a premature entry into mitosis. So he looked for other genes that gave the same phenotype, and he collected, I think, sort of 49 alleles of WEE1, if not WEE1 itself. And then the 50th one that he looked at turned out to be an allele of CDC2. In other words, CDC2 is a gene that you can either kill, in which case the cells don't divide, or somehow make hyperactive, in which case it accelerates. So this then became the focus of Paul's attention. And as far as he was concerned, it catalyzed entry into mitosis. Lee's gene catalyzed entry into S phase, so they looked like they should be very, very different. And it came as a total surprise when it discovered they were actually the same thing and interchangeable. Not only that, but from its sequence, it was cloned and sequenced, and it was found to be a protein kinase. So where does cyclin fit in? Well, it took a long time, and basically the heroes there were Canadian Fred Locher and an American Jim Muller, who succeeded finally in purifying MPF and discovering that it had two subunits, of which the small one was CDC2, the larger one was cyclin B, and if you assayed it for protein phosphorylation, it had powerful protein kinase activity. So that was the answer. And amazingly, you know, I've been working on this stuff for 5 years or something like that, and it never crossed our mind that we might be working on another protein kinase, even though I knew all about them. Here is Jim. He was tragically killed last year in a fatal car accident. I think killed instantly, actually. And he was a good pal, actually. Very, very nice man. So that's the answer. Here's CDC2 on the left, cyclin on the right, and basically CDC2 needs cyclin to turn it on. End of story. Problem solved. So once you understand everything, it becomes amazingly simple. It's extraordinary how foggy it is before you understand. So cyclin builds up, CDC2 turns on, and the cell enters mitosis. You degrade the cyclin, that's another story. CDC2 turns off, and the cell exits mitosis. And really, that's all there is to it. And so, you know, I was terribly lucky to be in on the ground floor and terribly lucky to do a sort of random experiment for the wrong reason that gave, it's a good example, I suppose, of chance. And you just never know, in my experience, what funny twists and turns, and it depends very much on, you know, where you are, what you're working on, who you're working with. people can be incredibly useful, or they can be absolutely poisonous in my experience, and that's the story. And I think the other thing is that, you know, it's really important to go for it. If you see a chance, grab it, because it may never come again. Thanks for listening. Sorry I've gone on so.